Welcome everyone to the DSORN booth at ADLM for this podcast titled The Sepsis Equation, Better Care, Smarter Economics. Today we are joined by Eran Eden, co-founder and CEO of MEMAD Diagnostics, and he was involved in pioneering the MEMAD BV test, which is a different way of looking at infection that focuses not on pathogen identification, but on how differences in the human immune response to broad classes of pathogens can be used to distinguish bacterial and viral etiology. This is the most important aspect of acute infection that drives the decision to administer or withhold antibiotics. Today we will discuss the clinical problem, the evidence supporting the MEMAD BV test, and how better diagnostic information can support better care and smarter hospital decision-making. So, Eran, please tell us about your background and what led you to start MEMAD many years ago.
Well, first of all, it's great to have this conversation. There's two stories of how we started MEMAD. I'll go for the second one. The second one, basically, I was doing my PhD. I have a background both in computer science, doing a lot of work on machine learning, and also as a molecular immunologist. And frankly, I got frustrated. Myself and another co-founder is an MD PhD, Dr. Kfir Ved, we got frustrated by the gap between scientific discovery and the ability to really have an impact on real patients. So we said, let's play a game. And the game was, well, what if we harness the power of, number one, the body's immune response, plus measurement capabilities, plus AI, at that time was not a sexy topic. And if we merge them together, what type of big, ugly problems that don't have a good solution in 21st century medicine could we go after? So it started just by this naive concept or idea. And at that time, again, we're doing our PhDs. We got a room or a kitchen to work in, which is Kfir's grandmother's kitchen. And that's where we started with this initial idea. And this idea then led to, I would say, a proof of principle, which led, God forbid, to a product and FDA regulation and reimbursement and health economics and many, many things. But it all started about 12, 13 years ago.
So this concept of host response is new to some of the listeners here. Can you describe what that is and how it can be used for this kind of test?
Absolutely. So the concept itself is based on something quite intuitive, which is that our immune system is a very eloquent system to tell us what's going on in our bodies. And if we just had a way to measure it really quickly and then incorporate all those signals into simple insights, that could have a pretty profound impact. And the first problem we went after is arguably one of the most prevalent medical encounters on the planet. On one side of the extreme, we have a child with sniffles. I have 3 of them back home. And on the other extreme, you have maybe an elderly patient with suspected sepsis, which is a life-threatening indication. And the idea there is to look at the body's signal, the body's immune response signal to these varying diseases, and incorporate that into different insights that can potentially help you trace the source of the infection, and then provide you with very meaningful information that can help guide treatment. So this is really from a bird's eye view. Double-clicking on this, maybe it's important to say what this is not. Because look, we live in a world where there's standard of care, there's the clinical gestalt, and there's direct pathogen detection tools that's been around for many, many years, like cultures, like PCRs, like rapid antigen tests, and they're good. And DSORN, I think, has a really interesting both repertoire of products and more to come. I know that you're right now launching additional things. So the idea is, how can we contribute? And why is the body's immune response really providing added information for what's already maybe solved today? So it turns out that there's several fundamental limitations of current pathogen detection technologies. The first, more often than not, no microorganism is detected, even if you use a multiplex PCR panel. But as a clinician, you have to make decisions 100% of the time. As a patient, you want to get a reliable information 100% of the time, especially if it's your kid. By using the body's immune response, what's called host response, you get an answer in the majority of the cases. So that's one differentiator of host response. Another is inaccessible infections. So for example, you can run a swab in cases of pharyngitis, if the infection is in your throat. But what about otitis media, ear infection, sinusitis, bronchitis, pneumonia? Try to get a sputum sample from an elderly patient that has pneumonia. That's very, very hard. Again, the immune response is not just a geeky nuance. It goes into these inaccessible infection sites behind your tympanic membrane in the lungs, and from the periphery, it tells us what's there. A third example, co-infections. So the fact that you detect a virus with a very accurate PCR doesn't rule out a bacterial superinfection. It's a huge issue. Influenza viruses tend to be the most friendly viruses. They bring their best buddies, pneumococcal bacterial superinfections, they throw out a party. So by using the body's immune response, you actually can separate from a pure viral infection to a co-infection. And then the last example I'll give is the issue of colonization. Going back to a kid, let's say you pick up a kid from the street, a random kid, and you sample their nasopharynx, you will detect in 50% of the cases a rhinovirus, whether they're healthy or they're sick. So mere detection does not imply causality. The immune response knows if that rhinovirus, as an example, is a bystander or if it's attacking the body. So using the body's host immune response is not just a geeky nuance for scientists. It helps you fundamentally change the paradigm and complement what's out there today.
So unfortunately, we only have 20 minutes in this podcast, but I do want to ask simply, how does the MeMed-BV test work? And what does the result mean?
The way that it works, you actually measure 3 circulating soluble proteins that are in the body. It took us several years to actually identify them from running perhaps one of the largest proteomic screening of the body's immune response, but eventually 3 proteins. The first is called TRAIL. TRAIL goes up in viruses and goes down in bacteria, and it's actually the first FDA-cleared biomarker that goes up in viruses. Interesting anecdote. The second, C-reactive protein, goes up in bacterial infections and in general inflammation, has been around for about 40 years. And the third one is called IP10, interferon gamma-induced protein, which goes up in any infection and higher in viral infection. None of them is perfect by themselves, but the combination gave us the best performing signature from all the millions of other combinations. So you take a small volume of blood, a few hundreds of microliters, on MeMed case 150 microliters of whole blood, on the liaison perhaps a bit more than that, and then depending on the instrument, you basically plug it inside and you get a score. The score is between 0 and 100. The higher the score, the higher the likelihood of bacterial infection. Low scores, no bacterial infections, so primarily viral infections. High scores, bacterial infections, you want to treat with antibiotics.
There's a whole field called test implementation science. How do you go from developing a new test through the process that you alluded to before, initial testing, publication, CE, FDA clearance, onto implementation? If you think back to the last 10 or 12 years, what were the key breakthrough moments when you remember as a PhD student, you find that you got a result when you said, I'm going to get my PhD based on that one result? What were like 2 of the key moments of excitement during that journey for you?
First of all, if we have to be very honest, most of the, I would say, milestones along the way were pain points with a few highlights. So I remember the pain was initially we couldn't find any biomarker that was better than standard of care. The highlight was when we found that TRAIL has this really unique property that it goes up in viruses and goes down in bacteria. And initially when we saw this, we said, this is BS. We didn't believe this. It took many years and others to actually reproduce the results before we actually believed. So that's one example of a eureka moment. Then another one, as you go through the process, you have to show that what you measure today and yesterday in my hands and in your hands are the same, what's called analytical performance. It took us a very long time to go through this. No eureka moment, just a lot of iterations. Another really important milestone was the clinical validity part. So the clinical validity is there, to show that you're accurate. Sensitivity, specificity, PPV, NPV, raw curves. We published the first study after about 4 to 5 years, and we were ecstatic about this. We even got a phone call from BBC telling us, listen, this is potentially transformative. but we don't believe a word you're saying because the field has been plagued with overpromise and underdelivery. You need external, not internal, prospective, not retrospective, double-blind studies, not the type that you do peekaboo, led by independent key opinion leaders, not a bunch of wise guys running around in Haifa and Boston. That takes a lot of money, a lot of time, and most companies fail. Good luck. So just to complete the thought there, we gave the technology to external folks that ran this for about 3 years and then unblinded the results. The first results were published in the Lancet ID, and they reproduced the same result that we got. And then we knew that this is something real. This was one of the biggest highlights.
So now flash forward after now dozens of papers by those independent scientists and clinicians showing that the test works. How do you go from that point to the point we're in now, where now people are asking, where's the best place for it? What populations would it work in? And what populations that might not be included in FDA clearance and so on might also be potential populations for future?
Great question. So the FDA clearance that we got was very broad and included children and adults, any suspected acute infection, upper respiratory, lower respiratory tract infections, suspected sepsis, urinary tract infections, centralized and urgent cares. And so the question was, where do you actually see the most value? Because you can't start everywhere. And together with our customers and partners, we identified 4 archetypes. The first, adults suspected sepsis. There's a lot of value and pain, both healthcare and health economic in the system, and we can talk about that. The second was adults and elderly patients with lower respiratory tract infections. The third was kids with lower respiratory tract infections. And lastly was kids with fever without sorts. They're basically crying in mommy and daddy's ear, and you have no clue what to do. These are 4 bridges to which we usually introduce a BV. And there's a different interplay if you do this in the hospital setting or decentralized setting, because there's so many nuances. But high level, this is where we're seeing a lot of the value and a lot of the uptake, and we can double click on what we're actually seeing there.
So the clinical utility of the test is the most important concern to hospital administrators too. It doesn't matter whether it's inexpensive to run and so on if it doesn't work. But let's face it, the economics are important as an important second question. So what can you tell us about the value of this in cost savings in whatever way that term means of this test?
Yeah, you're absolutely right. Showing performance is not enough. You have to show outcomes, clinical outcomes, objective clinical outcomes, and health economic outcomes. And this is something we've been passionate, borderline obsessed over the last 3 years. So first of all, where is the evidence coming from? It's coming from real-world evidence and randomized controlled trials that we've been doing together with you, which is very untypical of diagnostics. This is more of a pharma play, but we've actually taken the risk and the time and the capital to do that. What are we seeing? Well, the first part of the equation, the ability to rule out a bacterial infection with a very high degree of performance. By the way, we have a negative predictive value of 99%. That is, in multiple comparisons, even against experts, it's higher than the human capability. If you can do that, what you're starting to see is, number one, reduction of antibiotics, unnecessary antibiotics, in those cases where the tests tell you there's a non-bacterial case. As an example, we've been seeing anywhere between 25 to 60% antibiotics reduction, including IV antibiotics. So that's one element. But some would say that some antibiotics are cheap. So that's not necessarily going to be the driver, even though from a societal perspective, it's really important for the individual patient or the individual doctor in the hospital, that will not always move the needle. The second element that we're seeing, again, relating to rule out of bacterial infections, is reduction in length of stay. That is critical for hospitals. There's just been a publication on 1,500 kids before and after implementation showing a 0.5 day reduction in average. Why? Because those kids don't necessarily need to have IV antibiotics. The minute you don't have to hook them up, you can actually send them home earlier. It's both better for the kid, it's better for the patient, and frankly, it's also a revenue driver for the hospital. So that's a second outcome. A third outcome that we've just got published by a US network is preventing unnecessary admissions. 19% pre and post implementation, specifically in the population that had MMBV equals viral. So these are examples in the centralized setting. Decentralized setting, slightly different. So you see things related to uncertainty, patient satisfaction, but also we just had another implementation study by a 1.5 million member HMO that rolled this out nationwide, and they were showing potential avoidance of unnecessary referrals to the hospital, which also adds a very big economic burden to the payer, up to about 25%. So the same tool can give you different things. The last thing, and this is the surprise that we had. While we started this about antibiotics avoidance when it's not necessary, what we're starting to see more and more that we have cases of bacterial infections that we're preventing them from being missed. So I don't know how much time we have today. There's a few examples that have been published recently where this is saving lives and saving tremendous amount of cost just by preventing this missed bacterial sepsis that is avoided. And we have today the data to show that. So again, you run the show today. You tell me how much time you want to go there.
Well, we don't have time to get into those, but this is a nice segue to getting back to the title of the podcast, the sepsis equation. So I guess we better talk about sepsis a little bit. How can this test be used to manage someone with suspected sepsis?
Okay. So first of all, just to define sepsis, immune dysregulation of the body, where basically it's the immune response that's operating many times in hyperdrive. That's what's potentially hurting the body. And it's one of the lead causes of mortality in U.S. hospitals today. Now, the best way to treat sepsis is to not reach that situation from the get-go. Early detection. Now, this is obviously no single test will give you all the solution for that. It's a concert effect of multiple things, the clinical gestalt, sepsis order sets, bundles, what have you. Now, if it's a very severe case, frankly, you don't really need any diagnostics. You're going to treat them, and you're going to treat them very aggressively. If it's a very benign case, you don't need any diagnostic either. It's those in-betweeners that the guidelines are talking about trying to figure out within about 3 hours. That's where it's becoming really, really interesting.
And there are many of those patients.
Exactly. Hospitals are seeing many of those patients coming in and out every day. And making that decision, especially in a stressful environment like an ED, is non-trivial. Here's an example that just got published by Johns Hopkins. It's a use case. So they had a healthcare worker at her 40s, post-shift. She was fatigued and went to the head of the ED at the time, said, okay, just give me a regular checkup. Vital signs were not very dramatic. And they ran a MIMID-BV test. The score came out 100. Said, wait a minute. IV antibiotics, watchful waiting, stay here. She actually crashed after about 2 or 3 hours. But because she was already in the ward, she was put in the ICU, fluid resuscitation, and after a couple of days, she was released. Now, that is an example where maybe that patient would be treated, maybe it would be sent home. This definitely was a tipping point. So that's an example where this can really make a difference. Now, you could say, maybe it's a one-off. But what happens if you follow the data, you see that in our randomized control trials, in the control arm, there's cases where MIMID-BV is giving you a bacterial score. The clinicians are blinded to MIMID-BV. So they're sending those patients home, and they turn up back in the hospital after 24 hours with a working hypothesis of sepsis. And this was published as well. So this is not just anecdotal data. That is something where I think we have a potential to contribute quite dramatically.
So we're just about out of time here, but if there were one take-home message that you'd like to leave with this audience, which is a mixture of laboratorians, clinicians, and other interested people, what would that be?
Well, I think for laboratorians, I would say here is an opportunity to not only drive efficiency, there's a lot of data on that, but it's to impact patient lives. For clinicians, just reducing this uncertainty, often just helping your clinical gestalt, but sometimes giving you an added value, like extra advice that could potentially be quite meaningful. For the patients, I would say for the hysterical mothers, like my wife, well, this could help. But even more importantly, I would say watch out from sepsis. This is such a horrible disease. You have to have awareness. And then finally, to entrepreneurs, it's you climb a Mount Impossible for many, many years, eat a lot of rubble and barrels, but it's worth it. Eventually, it's worth it.
Thank you, Iran, for your insights in the MeMed-BV test, and as one of its founders, of course. And for listeners at ADLM, be sure to visit the DiaSorin booth and website to learn more about the MeMed-BV test and other testing offered by DiaSorin. Thank you.
Thank you very much.